So it's my great pleasure to very quickly, so we keep moving, introduce Dr. Shafali Jeste, who is our pediatric neurologist in chief in the autism world here. And she's been doing, not only is running with Dr. Martinez, what is now the Caring Clinic, the Developmental Neurogenetics, but also directs multiple studies that are trying to identify what we call biomarkers. And by the end of this, you'll know what they are.
Thanks. Okay, hi everyone. Hopefully you're not too tired after lunch. But in the past at the symposium, I've actually talked mostly about clinical and neurological issues in autism, and I'm actually taking a different angle today and talking to you a bit more about our research. And part of the reason for that is that I think that this symposium is a really nice opportunity for us to really share with you what we're doing in the moment that we really do hope and expect to have real clinical implications. And I think it helps all of us, for those of you who are clinicians who take care of kids with autism or parents, to be better informed consumers of the literature. And really my goal with actually having Dr. Jeste give that really, I thought, beautiful talk was just to help, I think, help all of us think about when we read clinical trials and we read that they've failed or that they've struggled or that there's no meaningful outcome, what does that really mean, right? What does it mean when someone tells you that a drug does work, right? What are the questions you want to ask of a practitioner or someone else when they're telling you that a drug is, you know, fixing a symptom, right? So all of this is really to help, I think, everyone become better consumers of this very, very, you know, vastly expanding literature on clinical trials. So quick disclosures here, none of which have bearing on the work I'm going to show you today. But, you know, I really give this talk and do this work from the heart of a clinician who sees patients every week who come to me trying many, many, many different alternative and complementary treatments that they've been told will cure their child's autism or will help, you know, dramatically fix a problem or a situation. And while, of course, we want therapeutics, and I am a part of this big group who's really working towards that, I think it's, again, really important for us to be mindful as to what is an effective therapeutic, and conversely, why do some of our therapeutic trials really, you know, struggle or fail? And that's really where our biomarkers research comes in. And so I'm going to start by giving you just some basic concepts in the term biomarkers, because it's a big buzzword in our field right now. So I think we'll just talk about some basic definitions. I'm then going to talk to you about the work we do in our program focused on EEG biomarkers, and I'm going to give you just quick snippets of research that we're doing across a few different studies just to sort of whet your appetite and get you excited. Each of these snippets could be a talk in itself, but I think it's a nice way to give you an introduction of what we're doing. And then we'll stop and we'll take questions at the end of the day. So you've heard a lot about precision therapeutics from Dr. Geschwind, and this is definitely an area of emerging excitement. We really want to be at a place in autism where we have targeted treatments that are directly affecting and improving the underlying etiologies that are causing our symptoms of autism or neurodevelopmental conditions. But unfortunately, even in targeted therapeutic trials, like this is an example of Fragile X, and this was a review that was done recently that reviewed all the trials in Fragile X, where there were clearly mechanistically driven treatments that should have worked, and you can see the list of all the different possible treatments and all the different outcome measures that were used. And unfortunately, none of these trials were deemed successes. Okay, so it begs the question, why do trials fail? And Charlotte already actually showed you kind of the rubric that I use a lot to think about that, and so we won't go belabor this too much. It might be that the drug just isn't working, it's missing the target. It might be that we're selecting the wrong group of patients. We're studying kids that are too old, meaning too late in their development, or maybe even too young, or that we're choosing a whole heterogeneous sample of kids when only a subset of those really would have been the right participants in that trial or more amenable to treatment. Or it might be our outcome measures, and this is the big sticky part, right? You've heard so many talks now about trials and outcome measures, and as you've seen, there's a lot of issues around the way we're measuring success. And this is where biomarkers research is really driven and inspired, right? So what is a biomarker? So a biomarker is any characteristic that's objectively measured and evaluated as an indication of a normal biological process, a pathogenic process, or a response to treatment. Okay, so we can probably think of many biomarkers in medicine, right? The classic example being something like blood sugar and diabetes. So an elevated blood sugar really is helpful to diagnose diabetes, and then when we treat the diabetes with insulin or other types of agents, we actually monitor the blood sugar as a response. That's incredibly concrete and super easy to measure, right? But let's start thinking now about how are we going to start thinking about biomarkers in autism. This is a lot more complicated. But before we get there, we can also just think about how are biomarkers going to address some of these issues that we've addressed. Well, from the drug standpoint, these biomarkers might help actually measure target engagement. Is the drug actually hitting the target? Again, going back to the diabetes example, if you give insulin and you see that the blood sugar goes down, that might suggest that it's hitting the right target, right? It also might help refine our selection of patients. If we have a biomarker, an objective measure that can stratify individuals in a meaningful way, then we might be able to say, hey, this group with a certain level of blood sugar, for instance, might be more amenable to a treatment. Provides an objective way to select patients for a trial. And then lastly, it could actually be a nice proxy for an outcome measure. Now, when I say this, sometimes people say, well, but our goal isn't to fix a biomarker. We'd like to improve adaptive living, right? We'd like to improve social communication skills. Our goal is not to fix a biomarker. But it may be that our biomarkers as objective measures of whatever the underlying process is, those actually might be good intermediate measures that tell us that the drug is doing what it's supposed to do, which then might kind of precede the clinical outcome that we expect to see. It might help us decide that we want to actually keep a drug ongoing or a trial ongoing because the biomarker has actually changed. Okay, so there's a lot of interest, and again, this is really where our field is at in terms of taking these principles and biomarkers and thinking about how they might apply to autism. We also actually need some other characteristics to be met in biomarkers, right? So we want them to be grounded in mechanism. We can't use blood sugar for autism because they're completely unrelated, right? We need a biomarker that is rooted in the underlying biological mechanism of the condition. We need them to be feasible and practical, something that we can actually collect, data that we can collect. Even if we first are doing it in the lab, we need to then be able to somehow scale it out into the community or somewhere where it's more practical. We'd like it to be reliable over time and place so that if you test it in spot A versus spot B, there's not a different result. So we need consistency. And then we'd like it to have what we call discriminant validity and stratification ability. So discriminant ability is, does the biomarker actually differentiate your clinical population from a non-clinical population? And stratification ability is, does it actually relate to some clinical symptomatology that's relevant? Does it stratify individuals in a meaningful way? And then lastly, I put it kind of in question marks, but I think it actually, maybe should actually be an exclamation mark, which is that we'd like our biomarkers to have some sort of translational potential, meaning wouldn't it be nice to have a marker in a patient that we can then translate back and measure in a preclinical model like a mouse model, so when we're doing drug discovery and trying to test drugs, we can see if that marker actually changes in that model. So let's think about now biomarkers for autism. So where are we going to look? Well, of course, we all probably agree that we need to look in the brain because that's where autism is essentially rooted. But how do we think about the mechanism? And this is where actually genetics gives us quite a bit of insight. And luckily, Dan already gave you a beautiful talk on this. So I can just say that what we've learned from genetics is that fundamental processes in brain development are affected very, very, very early. And they affect essentially two, I'd say, major kind of principles. One is the way that brain circuits are formed. And then the second is really the balance of excitatory and inhibitory circuits in our brain. But really, this is from a cellular all the way up to a systems and neuroanatomical level, really affecting basic systems in brain development. This was a term that actually Dan coined many years ago, that autism was a developmental disconnection syndrome. And I actually still use this because I like this as we root our work in EEG actually in this concept, which is that these connections in the brain are just not forming the same way they might be forming in typically developing children. So how do we measure this process, right? So this is sort of a challenging endeavor, but this is where our methodologies actually can be very helpful. And the methodology that we use in our lab and that many labs are using around the country and world is electroencephalography. So some of you may be familiar with EEG, a tool that we use to detect and diagnose epilepsy or seizures, and it's a very clinically available, easy tool. We use these fancy, expensive caps in our lab, in part because these caps are really really easy to put on, and we don't require individual electrode placement, but we're actually gathering the same data that we're gathering from those clinical EEGs that you see. So what is EEG really doing? So let's think of it this way. So when the neurons in the brain fire and communicate with each other, they generate these tiny little electrical signals. Okay, but we have millions and billions of neurons in our brain, and so when thousands of neurons fire in sync, that electrical signal gets big enough that we can actually measure it with the help of some amplifiers on the scalp. So the deflections that we see actually on the scalp are really reflective of this synchronous firing of neurons. And this is just kind of a cartoon to show you that, right? So all these neurons are firing together, and they're generating the signal on the scalp that we see here. So we can actually be quite creative with the way we process that signal. We don't make pictures as pretty as Morella's and Susan's in terms of imaging, but we learn some really, really beautiful things about the brain by looking at EEG. So that signal that you see up in that corner that some of you may have seen if any of your either patients or children have had EEGs, we can break that signal down into its component parts. And those oscillations are really defined by the frequency at which those waves occur. And each of those frequencies actually has really, on its own, very important value and importance in terms of its significance for neurodevelopment. So each of these reflect different processes in brain development. We are particularly interested in alpha band oscillations because those really reflect the integrity of really subcortical to cortical long-range networks forming in the brain from a very, very, very early age. So what we can do when we break down the EEG into those component parts is we can actually quantify how much of that whole EEG is essentially accounted for by those different frequencies. And that's what we call spectral power. We can also quantify basically the peak at which each of those frequencies occur. So we can identify when a peak emerges and what the frequency of that peak is. And that also has actually really exquisite developmental significance. We also actually then, based on actually just that activity in the brain, form connectivity networks and measure how well different parts of the brain are actually connected together. And one method that we use in particular is called phase coherence. So phase coherence really rests on a principle that if two regions of the brain are well connected, the oscillations, the brain waves are going to occur in synchrony, right? And you can see, that was on my tap in. So you can see on the bottom right here, when you have two parts of the brain that are in high coherence, they're going to be in sync here, right? Whereas if you have two parts of the brain that are not quite well connected, they're going to have very low phase coherence. Those oscillations are not going to be entirely in sync. So we leverage this basic principle with lots of fancy math to then essentially quantify how coherent signals are. And then, as I talked about with that peak alpha, we can also look at just when the peak alpha emerges and quantify the frequency of peak alpha. This is an example of just one baby's EEG from 3 months all the way to 12 months of age. And you can see this is just basically the slope of the EEG. This is the power, and this is all the different frequency bands. And you can see by 6 months of age, this alpha peak starting to emerge that really nicely emerges by 12 months. And then over later childhood, that peak just becomes later, and we can quantify that. In typical development, peak alpha relates very nicely to age, which is important when I show you one of our studies in a minute. So EEG, besides the fact that all our babies look so happy wearing our nets and are adorable, is, on top of being mechanistically meaningful, it also is actually practical, right? So it doesn't require a task if we're looking at resting state networks. It's pretty motion tolerant, so children can move to some extent, and we can still gather nice data. It's repeatable because we're not asking children to learn a task or that we're not actually examining learning. We can actually examine resting state networks over time. And it's potentially scalable. So if we can really maximize the information that we can learn from our lab-based EEGs, we might be able to then scale that information out to clinical EEG. And I'm going to show you an example of that at the end. And so in that context, our lab, which is really the biomarkers core of our center and our ACE, really works under two principles. One is that we can develop and apply rigorous methods in EEG to identify changes largely in neural connectivity and synchrony through the peak alpha and power in autism, and that these efforts will lay the foundation for biomarker development for clinical trials in autism. And we have three really big lines of research in our program, and some of you may have had children or clients who have been in our studies. I would bet you that some kids associated with folks in this room have worn one of our nets because we have lots of studies going on at once. We have three lines of research, and I'm going to give you snippets of each one of these. The first is that we believe that we can actually identify EEG markers in early infancy that precede autism. And this actually piggybacks really nicely with what Morella talked about earlier today. Secondly, we actually work on identifying EEG markers of, again, aberrant or changes in connectivity in autism itself, with the goal of stratification. And then finally, we can identify EEG markers that relate specifically to particular genetic etiologies, really going back to mechanism. Okay, so I'm not going to belabor this too much. I will just say I said EEG is really easy, it's feasible, motion tolerance, yeah, blah, blah, blah. But it's actually not that easy, and we do need to have enough signal to noise that we're not just measuring noise. So we take great lengths in the lab to actually maximize our data quality. And we have many published studies that have described our methods in detail. I'll just say that we have lots of training nets that we send home. This was a child in one of our studies who loved the training net so much she literally didn't take it off for about a week, wore it to school, wore it at home, but it desensitizes because it is sometimes an aversive sensory experience to wear one of these nets. We also take great lengths in the room to make it comfortable, and we also develop visual schedules for families so that the children are ready to come. I won't go into this in too much detail too, but I will say, and actually Charlotte was very involved with this, we also appreciated the fact that a child's behavior and state in the room while being tested actually affects their EEG signal. And so we found a way, we developed a scale to quantify the child's state in the room, and we found that that state actually absolutely affects their actual EEG signal. Kids with a better state had a difference in alpha power here than kids with a more agitated state, if you will. And that's really important when we're trying to make meaning of these signals and use them as biomarkers, right? We need to be really thinking about how state can affect the signal. And so based on this, we actually account for state when we analyze our data. And then I'll lastly say, and then I'm going to show you some of the studies. And again, this is important. I'm bringing all these things up, again, to sort of help you be consumers of this literature, because there are EEG studies that are done in autism with variable results. And I think that, again, we take great pains to ensure that what we are doing is as robust as possible so that the signal is meaningful. When we map connectivity networks, what we do is we take an unbiased approach. So we work with a fantastic statistician, and I have a research scientist in the lab named Abby Dickinson, who's really spearheaded a lot of this work as well, where we basically take this rich data set and we ask through, again, permutation testing and other statistical approaches, which are the networks that emerge that seem to really differentiate the group that we're studying compared to a different group, be it infants at risk for autism or children with autism. And we let the data tell us which networks are really emerging as meaningful. And then we can test those, again, in a forward manner in future studies. Okay, so in that context, I'm now going to give you some kind of like a quick data blitz of some of the work we've been doing, hopefully to just get you kind of interested and exciting in the work. So first, remember I showed you those three big research areas. So the first was babies. So this is actually the same model of the study that Morella talked about already. She talked about the MRI and eye tracking piece. Well, in that same high-risk infant study, we collect electrophysiological data starting at 3 months of age. And there's really been no studies that have shown differences as early as 3 months of age in high-risk infants, particularly not differences that relate directly to autism symptomatology. So the study is still ongoing, but we do have nice data at 18 months from the ADOS toddler module. And we're able to put kids into categories as to high and low risk for autism based on the ADOS toddler module. And what we find, to cut to the chase, is that at 3 months of age, these are children who actually are showing symptoms of autism. This is at 3 months. And these are children who are not. And what you'll appreciate is that it seems as though some of these networks are more connected, more robust in the 3-month-olds who go on to have autism. And it turns out that is, in fact, the case. They have hyperconnectivity, particularly in long-range networks. And the one network that really comes out is this left hemispheric network here. And we find, you can see on this plot here, that the children who are ADOS-T positive at 3 months of age show significant overconnectivity. Along the same lines, we've actually taken that same connectivity analysis approach in children with autism, school-age children with autism. And here we find also that children with autism, and typically developing children here, have significant differences in long-range connectivity. By the time they reach school age, though, the difference is in the other direction. So what we're seeing is reduced interhemispheric connectivity in our children with ASD. And I didn't go into this in detail, but it actually maps really nicely with some of the early imaging literature that shows this early kind of overgrowth and overconnectivity that maybe rapidly declines over development, where by the time children are diagnosed with ASD, there's actually really an underconnection in certain important brain networks. And we're able to actually map this with, again, our electrophysiological techniques, which is quite exciting. And again, this scatter plot just shows this difference. You'll note, though, and I always like to show data as scatter plots rather than bar graphs so you can see every kid's data. I'm sure the children with ASD generally are showing this lower coherence, but there's a range, right? So it's not that every child with ASD is distinct from the child with typical development. And so the discriminant validity, if you will, of this connectivity measure may not be incredibly robust, but it's starting to give us a hint, right, of where we want to start looking and start focusing when we think about these electrophysiological biomarkers. Now, connectivity didn't really stratify our children with ASD, but interestingly, what did was in the same study, we also looked at, we looked at alpha coherence, as I showed you. We also looked at peak alpha frequency, so the peak at which that alpha oscillation occurs. And just like we would expect in typical development, our typically developing kids, peak alpha relates very, very strongly with age, just like we would expect to see. Not in autism. In autism, peak alpha relates very strongly with IQ, nonverbal cognitive function. So peak alpha seems to be very, very tightly related to cognitive function in our children with autism, and we think might be a really meaningful stratification biomarker in this sample. Okay, in the last couple minutes, I'm going to give you the last data blitz, which is on looking at EEG and genetic etiologies of ASD. And we're particularly going to talk about one syndromic form of ASD called 15q duplication syndrome that actually others have already alluded to. This is a highly penetrant copy number variant for autism. And in the interest of time, I won't go into the details of the genetics, just to say that there's many genes in that 15q region that are very important for brain development, including several GABA receptor genes that are quite important for essentially regulating cortical inhibition. And we got interested in this syndrome because anecdotally and in a couple of case reports, it was reported that they had this change on their EEG, these fast oscillations that looked like patients who take medications that actually target the GABA system, things like Ativan or Valium, benzodiazepines. So it looked like they were taking GABAergic medications, but they're not. So we thought this might be an interesting electrophysiological biomarker of this condition. Let's quantify this. And that's exactly what we did. We traveled to several family conferences to actually collect electrophysiological data from these kids who are all over the country. And we also studied them at UCLA. And what we found, in fact, was that children with Dup15q do have a very distinct electrophysiological signature characterized by elevations in the beta band, so a little faster than alpha in the beta band. And this effect size was almost 2, which is quite high for electrophysiological studies. We since then have done studies to look at whether we can scale that with clinical EEG. And on clinical EEGs in these kids, we can quantify that beta peak just as robustly as we can with our research EEG. So that actually has opened the door for a new study that we're doing where we're actually quantifying this biomarker in kids who have had clinical EEGs around the country with this condition. Makes it much more scalable. We also have found that those beta oscillations, particularly the peak at which they occur, correlate very, very strongly with nonverbal cognition. So what does this mean? And I'm going to sort of end, I have 2 more minutes and then we'll stop, so sorry. Let's take this sort of precision medicine idea through a little bit. If we think about a patient, right, who has symptoms of what might be Dup15q, and we do genetic testing and we find that they have a 15q duplication. right? So we know that that leads to disruptions in cortical inhibition with this overexpression of these GABA genes. Well, electrophysiologically, we might find this signature. We will find this signature of DUP15Q. Now let's fast forward and think about some drugs that are being developed, have already been developed, that target the GABA receptor or GABA antagonists. Well, in a clinical trial, we can use those beta oscillations as a marker of target engagement. If that drug is actually targeting the GABA system, we would expect those beta oscillations to be reduced, we hope. And it might actually also, since it seems to relate to nonverbal cognition in these kids, might also be a meaningful outcome measure. So this actually has opened the door to a couple of upcoming clinical trials in this condition. And it really was all perpetuated by our quantification of this biomarker. And so that's been a really exciting movement in this space where we actually are opening the doors for clinical trials through some of this work. Okay, so in summary, EEG serves as a promising method to study autism biomarkers. We found aberrant connectivity, peak alpha that stratifies, and that EEG power that can differentiate different genetic subtypes. Continued efforts are really going to determine if these measures can guide patient selection in trials and can help improve outcome measures. Our next steps are that we need collaborative efforts across sites, right? We can't do this as single sites alone. We also need to directly incorporate these biomarkers into trials, and that is actually some of the work we're doing in our current Autism Center of Excellence study. And then we need to scale these out into the community. And so my last slide is I'll just tell you that there's a big national effort right now called the Autism Biomarkers Consortium for Clinical Trials. This is an effort across multiple sites to do what we've been doing essentially here at UCLA across multiple sites. So you want to identify electrophysiological biomarkers in autism. It's an exciting study that includes kids ages 6 to 11 at 3 time points. There's no trial per se. We're just evaluating biomarkers for their stability and for their ability to stratify kids within the autism spectrum. So that's an ongoing study right now if anyone's interested in hearing more about it. And with that, I will stop. Thank you very much.